All right, very good morning there. Today we're also going to have a lecture on protein quality. We got also to understand amino acid pools. So by the end of this lecture, you're expected to define amino acid pool and appreciate its value or importance. Then define the protein turnover, understand nitrogen balance and protein quality, and then you should be able to measure proteins. Should be able to measure proteins. Yes, you should be able to measure protein quality and demonstrate the matrices that are used for measuring quality of these proteins. You should also be able to appreciate acids, bases, and buffers for serum protein measurements, explore the different methods used, their principles and their applicability in this lecture. So to start with, one of the most important thing here is amino acid pool. What really is that we have a lot of free amino acids that are present throughout in the plasma, the cells, and the extracellular fluids. So everywhere in your body, the amino acids, there are about 100 grams of free amino acids in dynamic state that are constantly replenished as they are utilized. Meaning there is a form of amino acids that are throughout in your body. That is what we call the amino acid pool. A certain collection of amino acids in your body in all compartments, be it in the cells, the plasma, extracellular fluids, all of them have some amino acids. So that is basically what we call the pool, and that's up to around 100 grams that are always in dynamic state, meaning they keep moving from one compartment to another and all that, and they are also utilized and replenished. The body can use them and also replace them, so they continue being 100 grams and they are maintained at that. So let's look at this side where there is an image where we have some green things. There is what we call the amino acid pool. Whatever you're seeing in blue is one that replenishing is like replacing or adding onto it, and then this other one that seems to be grays, the utilization of it. I'll get to explain. This other blue side here is how it is being reduced or utilized in your body. So let's get to understand that this very thing, this very amino acid pool is never a reserve, and actually your body does not have amino acid reserve or stores, just like literature says. So here what we are seeing is that one, there is digestion of dietary proteins, that it adds up to about 70 to 100 grams per day. And then there's synthesis of non-essential amino acids in your body. We've seen process of protein synthesis. We have seen how transamination occurs, that some amino acids are synthesized in the body. Then there's degradation of all the body proteins. Look at your red blood cells, they can be degraded, your muscle cells and everything, all those can be broken down and also add into the amount of amino acids in the body. However, we have other mechanisms that the body also utilizes this amino acid pool. One is during the process of protein synthesis. They use up to around 300 to 400 grams per day for protein synthesis, meaning your body is making new proteins each and every day. And this possibly is increased when you have damages, injuries on your skin and everything, so that new proteins are made from your body. And then there is synthesis of non-protein nitrogenous substances. These are called porphyrins, the creatinines, the purines, pyrimidines, niacid, thiacid, and hormones. Hormones contain nitrogens. Let's talk about the purines and pyrimidines that we discussed earlier under the process of protein synthesis. They are very important. So your body have to synthesize them also, and they utilize these very amino acids to make them. Then there is a catabolized in the liver. The liver breaks some of this thing down And one is for energy. There's also ketogenic fat, and then glycogenic glucose. So that is how the amino acid is being replenished and amino acid is being utilized. The amino acid pool, sorry. So a question can come here. Let's say explain how the amino acid pool is replenished or and utilized. Or then they can give you 5 marks or 6. Yeah, so that each of these points brings out a mark or a key point. So get to understand that. What is the importance of amino acid pool is that one, it allows high turnover of amino acids in a way that there's a functionality of your amino acids. You can easily utilize them. You have a lot in your body, or you have, it's more like helping you since you don't have a reserve. You can utilize your protein always, all the time. There is nothing like a deficiency at some point, because that pool helps you to get the amino acids from there, and then it gets replenished. So amino acids are made available and deficiency does not occur immediately. So when you look at some of these protein deficiency conditions, like the brittle hair, sparse hair, the sparse and thin hair, when they're brown and everything like that, the breaking nails, those things don't occur overnight. They don't occur immediately. It's a kind of deficiency. It's just that your body has the pool that is able to provide until it is actually getting so depleted and your body is getting less. However, when there's a negative balance, that's when you set in those symptoms of protein deficiency and everything. So it is constantly replenished by dietary proteins. As you eat your food that is containing proteins, it is replenished. It is added on that. So let's get to what you call protein turnover. This is most proteins in the body are constantly synthesized and degraded to maintain health. So old cells can be destroyed and then new cells being formed to keep you healthy. So in healthy adults, the amounts of proteins in the body is a constant, meaning the rate of protein synthesis is the same as the rate of the degradation. The rate at which your protein is being made and the rate at which it is broken down is the same, and that's what we call the protein turnover. A protein turnover is basically the measure of how much protein is being synthesized, how much is being degraded in your body. So it has to be a balance there, that your body synthesizes some and it degrades like exactly that. So this that is actually equivalent to 300 to 400 grams of proteins per day that is involved in this. Yeah. So not in a healthy well-fed individual, the amino acid pool is said to be in a steady state and the individual is said to be in nitrogen balance. Meaning this amino acid pool being in a steady state is that it is replenished at the constant rate as it is being utilized. If you use 300 grams, you replenish with 300 grams. That is basically what we call the constant one. That is what we call the steady state of this amino acid pool. Let's understand what we call the nitrogen balance. Now, nitrogen balance, this is a comparison between nitrogen intake and loss from the body. We get to understand this, that you take in nitrogens in different forms, most especially in form of your proteins. The protein is nitrogenous, so anything that you take in that has proteins adds on the nitrogen content in your body. So what happened here is that to have a balance, you add in through intake of these foods, especially from your diet. But then there's also a factor that you lose them. One is that you can lose this through urea and then uric acid. As you sweat, as you urinate and everything, you're losing nitrogen. So that has to be always, there has to be a balance between intake and loss. Whatever you take in and whatever you have lost should have some kind of balance. So there is a kind of balance that we say. So one is therefore in the nitrogen equilibrium when the dietary intake is equal to the dietary loss. that is possibly in a healthy individual. If you're healthy, the amount of nitrogen you lose is the same as the amount of nitrogen that you take in through your diet and everything. But then there are other things that we call the negative nitrogen balance and positive nitrogen balance. Yes. That one, we have a positive nitrogen balance, meaning the intake of nitrogen, that I there means intake, is greater than the loss. Meaning you take more of this nitrogen, but you lose little of it. And this occurs in scenarios of growth, where your body is truly growing. Let's look at small children that are rapidly growing. It's more like they have a positive nitrogen balance, meaning more of this nitrogen they take into their body ends up being utilized in synthesis of proteins and using them very well. And they are not excreted into urine and everything. You need to understand the difference between the amino acid pool and nitrogen balance. That here we are not losing, in amino acid pool, we are not losing anything. We can get also a difference between protein turnover and nitrogen balance. Protein turnover is the amount synthesized and amounts degraded. Here we are talking about the amount you take in terms of intake, dietary intake, and then the amount you lose in forms of uric acid and urea in your body. So here, when you talk about the positive nitrogen balance, it occurs in a way that one, it occurs in growth, that most of these amino acids or nitrogen molecules are utilized in synthesis of protein in your body. And then second is that it also occurs in pregnancy, where the whole body of the woman needs a lot of proteins. You know what happens physiologically in pregnancy is that a pregnant woman will need more nitrogen or proteins, amino acids, to build up her own body and also maintain that of the fetus that is growing. And the second is recovering from illnesses, maybe injuries also. You will need more of this. So you will take in a lot, you will lose less, and much of it will be utilized for the process of recovery. Then hormones, we have growth hormones, the insulin and androgens. These are also some of those things that ends up increasing the utilization of these proteins for the different mechanisms instead of losing them. So you end up having a positive nitrogen balance. However, there's what we call the negative nitrogen balance, where we have your intake is less than the loss. That we have in other things like the chronic or acute illnesses that you have. And then you have starvation, where your body is utilizing a lot. Your body is losing a lot during that process, and you're not actually taking in more. During proteins deficiency, you also have an instance of taking in, meaning you took in less and you have lost a lot of proteins in your body, so there's no balance in that. Then the hormone corticosteroids also. Let's look into a concept of protein quality. And there are 20 common amino acids that are composed in the protein. And one of them is they are grouped into the non-essential amino acids. And these are amino acids that your body can synthesize. Please name them. Your body can make them, can make them for themselves. And then the essential amino acids here are those that can not be synthesized by the body, but they must be supplied from your diet. Your body has to make them. So with that, we also get to understand other amino acids. We also get to understand that the other amino acids called the conditionally essential amino acids. So what happens is that these amino acids become essential under certain conditions. For example, tyrosine is synthesized by phenylalanine, and if diet is low in phenylalanine, then tyrosine becomes conditionally essential. So those are basically what I can tell you. about these amino acids. So meaning these ones are actually the tyrosine is a non-essential amino acid and phenylalanine is essential. So what happens if you don't have phenylalanine, which is an essential amino acid that your body cannot synthesize? You're supposed to take it from diet. Then the tyrosine becomes an essential amino acid. So let me try to give some examples of these amino acids, starting with non-essential amino acids. We have the alanine, asparagine, aspartate, glutamate, glycine, serine, tyrosine, which is right here that we had mentioned earlier, that is a non-essential amino acids. And then the essential amino acids here, the arginine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, valine. Those that are conditionally essential amino acids are the cysteine, glutamine, hydroxyproline, proline, and taurine. Taurine is one of the rare amino acids that we have never heard about. You rarely hear about it, though it's also an amino acid. So those are some of the examples and some that happens or we have under these groupings of the amino acids. So the quality of dietary protein is a measure of its ability to provide the essential amino acids required for tissue maintenance. Now, how do you know a protein is quality? If it can provide for you most of these essential amino acids that you need. How much of these proteins can I get? So not all proteins are equal, meaning they differ in amino acid compositions, and their nutritive value also varies. In a way that you cannot say the meat is having the same nutritive value as beans. You can't tell me that fish is having the same nutritive value as genets. So you get to see that there's differences because of the amino acid composition. Some of these really have a lot of these essential amino acids in them that your body, some of them have a lot of these essential amino acids that your body cannot synthesize, but you have to take them from your diet. So that's what protein quality is, that you measure the ability of a certain protein to provide the essential amino acids that are required for tissue maintenance. Now, the quality of a protein can be demonstrated through a number of matrices that has been used. One is, as we have the amino acid score, which is also called chemical score or amino acid chemical score. We have the protein efficiency ratio, biological value, which is we're also going to discuss about it, and then the net protein utilization. Then up next is the protein digestibility corrected amino acid score. We'll also get to understand what they are. So to start with what we call the amino acid score, which in other words is also called the amino acid chemical score or just the chemical score, it's all the same. So this amino acid score provides a way to predict how efficiently a protein will meet a person's amino acid need. So this concept assumes that tissue protein synthesis is limited unless all required amino acids are available at the same time and in appropriate amounts at the site of tissue protein synthesis. Let's look at this, that your body wants to make a certain protein, but then an amino acid is missing, meaning your body won't make it. So what happened here is that this amino acid score tells us how efficiently a protein will be able to meet a person's amino acid needs. Meaning, if you need to make a certain amino acid, can this protein provide for you all the amino acids you need to make your proteins in your body? So that's basically what you call an amino acid score. Works on the assumptions that the tissue protein synthesis is limited, meaning it can't occur efficiently or as expected, or actually it can be halted unless you have all the amino acids that you require at the same time and also appropriate amounts at the site of tissue repair. If I'm making collagen, and then one of the amino acids required for collagen synthesis is not available, then definitely I'm not going to make collagen. Or if it's available but it is not obtained by the body at the same time, that maybe at a given point I have all other amino acids apart from one, and then the one comes in, or my body gets it maybe after some time or after some few minutes or hours, then the process of protein synthesis, collagen synthesis, will have to first halt or stop at that point, or it can be limited by all these variations and changes. All of them should be available at the site of the synthesis. Once I'm synthesizing collagen at a particular site, all its amino acids that are required should be available. Once one is missing or one's come in or the body obtains it later on, the process does not occur appropriately. That's just an assumption of what the amino acid score. So this helps us to predict how efficiently a protein will meet a person's amino acid needs. What amino acids do I need, but can the protein that I'm taking give me? Look at eggs, can it give me? Look at meat, can it give me? Milk. We look at also beans, can it give me all the amino acids I want if I eat beans for me to synthesize a certain protein? No, obviously. So if it can't, then you score. What is the score? What's the percentage? That's what we call amino acid score. So the amino acid content of a food being evaluated is compared with that of the reference protein known to be high quality. Of high quality. For example, egg protein. Egg protein is one of the one of the foods that is known to be of high biological value, meaning it contains all essential amino acids that your body cannot make, it will provide it. So what happened here is that you look at, you evaluate the amino acid content of one food, you compare it to the reference protein. Let's look at an example here that I have beans. You find out what is the amino acid content of beans, and then you compare it to the reference proteins of egg protein, which we already know is of high biological value. It has all the essential amino acids. Now you compare it. That is what we call the amino acid score. That what gives you amino acid score. Now, example, if lysine content of a whole wheat flour protein is 2.6% and the value of lysine in the ideal protein is 5.1%, amino acid score is calculated as 2.6 over 5.1 times 100, which is 51%. That's the formula for amino acid score. This lysine actually is absent in beans. Let's say we are comparing it to our reference protein we talked about earlier, that is an egg. We say the content of lysine in beans is 2.6%. And the content of the same amino acid lysine in eggs is 5.1%. What is the amino acid score? You just feed in everything into the formula that amino acid score is equals to essential amino acids in 100 grams of the test protein, which in this case is beans. And this essential amino acid, we are looking at, let's say the amount of lysine in 100 grams of beans over the amount of lysine in 100 grams of egg protein times 100. That's how we feed in everything. So the essential amino acids with the lowest amino acid score for certain protein is what we call the limiting amino acids, meaning it's one that limits everything. The essential amino acids with the lowest amino acid score, when everything, all you've done in the beans, you have all amino acids that have the high score and everything, and they're all available. But all of a sudden, this lysine with 2.6, then lysine becomes a limiting amino acid, meaning it has made the food not to become a complete protein. So let's go about this drawbacks or challenges with this type of metrics for measuring protein quality is that amino acid score is determined after acid hydrolysis of protein, which results into total amino acid content. So you can get this unless you break down the protein. And then you might end up to measuring the total proteins in everything. Second is that some of the amino acids in the total pool are not bioavailable. You may measure an amino acid and yet some of them are not usable by the body. You've heard some of these rare amino acids story. The degree of error varies depending on the food anyway. And those that are highly processed food has an impaired digestibility. That's why most of these missionaries would just tell you to eat whole foods and everything. However, when the food is processed, their digestibility is reduced or impaired. And then proteins with plant origin have a lower digestibility. That's why if we are going to look into biological value, that mostly those plants or those foods with high biological value are animal sources compared to plant sources. So the digestion of a plant protein is harder, or it is lower. It is not really that so much efficient. And let's look at the next matrix, which is biological value, and it is long considered the method of choice for estimating nutritive value of proteins. That has been considered as the standard long back then. And biological value of a dietary protein is related to the extent to which they provide all the necessary amino acids. So we can basically define biological value that the way you see it here. The extent to which a protein provides all the necessary amino acids that are required by the body. So let's look at biological value is the same nitrogen retained in the body, and theoretically is used in tissue and muscle formation, divided by the total amount of nitrogen absorbed from dietary proteins times 100. That's a formula for it if you want to find the biological value. The biological value is equals to nitrogen that has been retained in the body and being used in tissue and muscle formation, divided by the total amount of nitrogen that has been absorbed from dietary proteins. How much did you eat? How much did you eat, and how much did your body keep and used for tissue and muscle formation, and you multiply that by 100. So the values are obtained by measuring the fecal and urinary nitrogen, meaning you get those ones that you've lost. How much did you take in and how much did you lose? You'll be able to know how much has remained in your body for tissue one. You'll be able to know how much of this have been absorbed from your dietary intake. Yeah. So, and that when the test protein is fed and correcting for the amounts excreted when the nitrogen-free diet is fed. Each food has a different biological value, with some examples here that the egg has up to 94, milk has 90, and rice with 83, quinoa with 83, fish and beef and everything around 70. All wheat is 64, meaning wheat, much as is also known to have some to be a carbohydrate source, it also has gluten as a protein. We have beans, which is 58, lowest score. And then the next matrix for also measuring protein quality is the net protein utilization. That not very much different from biological value. This is not so much different, but we'll look into it. It measures how much of the protein in the diet is retained for use by the body, especially protein synthesis and tissue growth. You realize that they are talking of similar things. So this is determined by comparing the amount of nitrogen retained in the body with the amount of nitrogen eaten in the diet. How much did you retain in the body and how much did you eat from your diet? So in this case, by determining the difference between body nitrogen content of animal fed and now fed on protein, not I think that's a typing error. It's supposed to be fed on protein and those fed on a test protein. So you feed some animal we know experiments are done with animals. You don't start exactly with humans. So you feed some animal with a certain protein, you feed them on protein, and then others you feed them on a test protein that you want. And you feed some on eggs, some you feed them on beans, which becomes your net proteins. So net protein utilization here is that we have the total body protein at the end of the feeding trial, which is PB, minus the total body protein at the beginning of the feeding trial, meaning you know how much protein someone had before feeding, and then you give that person that protein, and then at the end of the feeding, how much protein that person have. The amount of protein consumed over the feeding trial, how much did this person consumed, you multiply it by 100. Take note of these formulas. There can be a question that you can calculate this thing. Then there is the protein efficiency ratio, and this is calculated by comparing weight gain of growing animals fed on test protein with the weight gain of those fed on reference protein. If you look at this, that we are still going to use the eggs and the beans example, that you have one animal you fed on beans, another animal you feed on eggs. Can that happen? Can animals feed on eggs really? So we can use human beings, that one human being has been fed constantly on beans, and another one has been fed on what? On eggs. So you compare the weight gain. Someone who ate beans, how much weight did you gain? And then someone who ate eggs, how much weight did that person gain over the same period of time? So with this method, there's no allowances made for maintenance. Method assumes that all protein is used for growth. So nothing like maintaining tissue repair and everything. All we know is that all proteins goes to increase your growth, make what? Increase your weight as well. So that was that. And then we get into what we call the protein digestibility corrected score, amino acid score, PDCAAS. It is the current gold standard for protein synthesis. Biological value being one of them long ago. Currently, it's with the protein digestibility corrected amino acid score. Is that it measures protein quality based on the human essential amino acid requirements and ability to digest it. Your ability to digest the particular protein, and that protein meets the requirement for your essential amino acids. So the test protein here is compared to the standard amino acid profile and is given a score from 0 to 1, with a score of 1 indicating maximum amino acid digestibility. So let's look at an equation here, that the milligrams of limiting amino acid in 1 gram of a test protein divided by the milligrams of same amino acid in 1 gram of the reference protein times the fecal true digestibility percentage. So what is this? True digestibility here is equals to human digestibility and fecal digestibility times 100. So the apparent net digestibility here, initial and final divided by initial times 100. True protein digestibility is the intake nitrogen intake minus into bracket the fecal nitrogen output of the test diet and the fecal nitrogen output on a non-protein diet. So someone is fed on also a non-protein diet, and then you divide by now our nitrogen intake times 100. That will give you the true protein digestibility. Let's get to understanding what we call the acid bases and buffers. Here is that the acids are one that donate hydrogens, and then bases, they accept hydrogen ions. Get to understand one of these key terms also, pH, which is a measure of acidity or alkalinity that has a pH scale of around 0 to 14. And then a buffer is a solution that maintains a constant pH, which in this case is our protein or amino acid. We are not looking into the chemical kind of buffers and everything, but we are going to focus on amino acids as a buffer solution. So a lower pH is more acidic, and when they say there's a higher pH, meaning it is more basic, and the neutral pH here is 7. Basically, amino acids and bases are chemical substances known for their constant contrasting relationship with hydrogen ions. Just like we said earlier that there are contrasting interaction with the hydrogen ions, that acids releases the hydrogen ion and bases accepts the hydrogen ions. So that is what this statement actually means here. Acids readily release their hydrogen ions, making the solution acidic, and then the bases snatch them, meaning grabbing them, gets them, accepting them. Yeah, now accept the hydrogen ions, then they create a more basic environment, meaning they reduce the acidity of the given environment, internal environment in most cases. For our case here that we are discussing about proteins. So the proteins are amino acid and a buffer effect. Let's look at this. That proteins are made up of amino acids. We all know that. And then an amino acid has an amino group and a carboxyl group, and therefore it is called a zwitter ion and also a side chain. What's a zwitter ion? Is a molecule that has this amino group and a carboxyl group that's equally charged. So they are all charged. So, and I can ask what a zwitter ion is, and then you give a definition of that. And then the amino and carboxyl group can ionize depending on the pH, and side chains do have diverse properties contributing to protein structure and function. So, basing on the acidity and alkalinity, that's determined by your amino acid or the carboxyl group ionization. And the structures of amino acids, the properties, their functions are determined by their side chain. By the side chain. Then amino acids and carboxyl group can act by changing their charge based on the surrounding pH. If its surrounding pH is alkaline, carboxyl group will ionize and release hydrogen there to neutralize it. And if it is basic, if it is acidic, the amino group will accept the hydrogen ions. So let's get to understand buffers and how they maintain a balance. So the amino and carboxyl groups of protein act as buffer groups, meaning they can accept and donate in response to the pH changes, and this helps to minimize some drastic changes in pH. So it is crucial for protein structure and functioning, given the fact that we know factors that affect protein structure, one of it being pH. So keeping that pH constant or within check is what maintains the structure and functioning of these amino acids. So those versatile amino acids and carboxyl groups of protein here are the ones that helps in all this process. So they try to grab in the excess hydrogen acid when when the plasma gets too acidic, or they try to release them when the environment becomes alkaline. So that's basically it for buffers. Let's continue. Now, when they become too basic, this remarkable ability helps maintain a stable pH around proteins, safeguarding their delicate structures and ensuring they can perform their essential task flawlessly, like I said earlier. Their functionality and properties is affected by pH. So this pH has to be kept at a stable rates so that they can really have their functions and structure in shape. So the isolation point, one of these key things to know is a specific pH where amino acid achieve a state of equal neutrality, meaning the pH is that pH where the amino acids, the positive charge is the same as the negative charge. That is what we call isoelectric point. There's some pH that makes it that way. Let's get to understand how we measure proteins. How do we measure proteins in our body? One is that we know serum protein play a vital role in maintaining our health. So we need to measure them. Measuring these will help. Measuring their levels offer a valuable insight into various disease states. So once you know the level of proteins, it gives you an understanding of certain diseases. Let's look into this. Let's delve into this. That serum albumin can communicate something. You find a certain amino acid or amino acids in blood, there's something that it communicates. When you find out that the albumin levels are changed from normal, it communicates something. So that helps you have an insight and into certain health disease conditions that are related to protein. And now this presentation is going to delve into this. So let's look at this. Why should we really assess serum protein? Is that one, if you assess the liver, it helps you to assess liver function using albumin and globulin ratio. And if that ratio is not normal, that can affect that can give you an indication that there's a challenge with liver function. Then you can diagnose inflammatory conditions. That is the C-reactive protein. This is one of the protein whose levels increases in your body when there's an inflammatory reaction taking place. So this, when there is a high levels of the C-reactive proteins, then that can indicate an inflammatory process taking place in your body. So we get to understand that. And then monitor kidney functions, albumin levels. When you start saying that your albumin levels appears in urine, you get to understand high levels of albumin in urine. You can get to have a deduction about that. Then it can also help you track nutritional status. Once your albumin levels are low or when they are too high, you can now understand that your nutritional status is quite unwell or it's not really stable or as expected. So what are the methods that we use for measuring protein? Is that one, we have the Biuret reaction. And what principle does it work on? First, we need to understand that it uses copper sulfate. So what does it do? That the principle is that the copper sulfates will react with a peptide bond in proteins, and then they form a violet complex. Now, that is exactly what it is. Then, after forming this violet complex, what next is that intensity of color is going to correlate with the protein concentration. The more intense the color, meaning the more the protein in your sample. So take an instance that I've taken blood and I add there copper sulfate. The reaction will take place that to form that violet complex. So that violet complex, someone else may have a very thick or high intense color of the violet. Someone may have just light violet. That gives a difference. So how is this applied is that you apply it in initial screening and also in overall protein status. If you want to find out how much protein status is in your body, how much protein do you really have or you're getting deficient, you can use this Biuret reaction that uses copper to sulfate to give you the results and understanding or an insight into your protein levels in your body. So it works on take note of the principles that this works on a principle that copper sulfate reacts with the peptide bonds in proteins and forming a violet complex. So you get to measure this, that you measure the color intensity of this protein and then you get your deduction about this reaction or the levels of proteins in your body. The total protein measurement provides a broad overview though, and the Biuret reaction relies on interaction between copper sulfate and the peptide bonds in the proteins. So this will generate what we call the colored complex, which is violet in color. The intensity of this color here is measured using what we call a spectrophotometer. And you put it on a spectrophotometer that will now help you determine how much exactly. Okay, it is intense, but what is that intensity? So if it is a negative test, the sample will turn into blue, given the fact that copper sulfate is blue. It will just turn blue, meaning there's no reaction that took place. But if it turns into purple or violet, that would now indicate that there are proteins there. So this can give you an insight. If I take my sample and they measure, it turns blue, meaning my blood has no proteins. If they take it and they measure and they find that they put the drop of the burette reagent, which is copper 2 sulfate, and it turns into purple violet, meaning I have proteins. Let's look at urine, where you can deduce kidney functions. Once this thing has been done and it turns into violet, they will understand that you have something related to your kidney function. And after that test tube that has been used, one gave you a positive test. Its test is taken and measured on a spectrophotometry to determine how much really is the protein that is found in your urine. So that's basically it about that. Another method is called electrophoresis, and it works on the principle that proteins will migrate in an electric field based on their size and charge. We get to understand this, that proteins move, yes, but they have a certain level of their charge. Some has positive, some has negative. Some proteins are bigger, some proteins are small. So there's a way how they separate them into different fields or brands or strands. So this separation is that it separates albumin and globulins into alpha, beta, and gamma. So they specify, it is important in specifying this is specific proteins. So you want to find gamma globulins, alpha globulins, it can separate them, all of them. So how is this applied is that it is important in identifying protein abnormalities. It monitors specific conditions. Electrophoresis work by applying an electric field, meaning that's based on the charges, and then separates them based on their size and the charge. Those with the big and positive charge, they go somewhere. Those with the big, small, negative charge, they go somewhere. Those who are just small, positive, and all that. They separate exactly based on that. And then the results gives you a visible bands, and each representing a specific protein fraction. Meaning, let's look at this structure here, this illustration this illustration here. That we have albumin being measured. At the band 1 we have one alpha albumin, we have alpha 2 albumin, beta albumin, and then the gamma albumin. It helps to separate. So this process of electrophoresis helps separate each of these proteins into specifically what they really are. Yeah. So this is important because it allows for identifying abnormal protein patterns. You find out that they are more of a certain protein than the other. Like inflammations, you have an increased gamma globulins. It detects. It will show you that you have inflammations in your body, and even detecting specific protein markers for certain diseases. So this is very important. Then let's look at the next method, which is an enzyme-linked immunosorbent assay, ELISA, or you just say immunoassay method. And it works on the principle of antibodies specifically binding to target proteins. And antibodies are specific in nature mostly, and they bond to a particular antibody. They bond to a particular protein and everything. So what happens is that how do you detect them? You say the enzyme links, the linked enzyme will generate a signal which is colored and is proportional to protein concentration. So that concentration of the intensity of that color changes is going to be measured and gives you an approximation or exact value of what it is. Where does it apply? It is used in measuring individual proteins like albumin. You want to find albumin, the C-reactive protein, which is CRP, and then specific disease markers. You want to find out what are some of these diseases have certain things, certain indicators. Biomarkers, some of these proteins are biomarkers. So it is a high specificity and sensitivity, and it is ideal for studying specific proteins of interest. Meaning this is highly sensitive and specific. It specifies the protein and sensitive, even meaning a minute level of the a certain protein in the sample can be detected, whether 0.000 something, it will still detect it. So that's basically what the enzymatic reaction is. And right here is that immunoassay offers a pinpoint accuracy. It is very accurate, and these techniques employ highly specific antibodies. So they use antibodies that are very specific and they bind to the particular protein. Need antibodies, they get to bind to our protein, maybe albumin, and then they form a reaction or change, and then that color intensity is measured, and we get to know what it is written. So the color intensity now will correlate with what? With our protein concentration. Thank you very much. I think we have come basically to the end of this lecture of ours today. And thank you.